Hi, I'm Chance.
And I'm Sarah Catherine.
And this is Conservation Connection,
presented by Last Chance Endeavors.
We run a wildlife education nonprofit focused on connecting students to their environment.
Each week here on Conservation Connection, we do just that by introducing you to the groundbreaking science and conservation work that's happening every day around the country.
We talk to professionals in the world of conservation science and wildlife management and ask them about their career,
their current projects,
their wild and crazy stories from the field,
and everything in between.
Join us each week to discover just how these dedicated people are working to protect our planet. Today, we're talking with Dr. Jody Beers. She's an assistant professor of biology here at College of Charleston.
Welcome to the show.
Thank you so much. It's great to be here. I'm really excited to do this interview with you today.
Absolutely. We're so happy to have you here. So could you give us kind of a background? How did you get to College of Charleston?
Yeah, absolutely. So my story on the path of coming to Charleston started about a year ago, right now, actually. When I first interviewed, I was super excited by the position that was opening up here. They were looking for sort of an integrative physiologist that did environmental science as a major focus. And I was super, super happy to put in an application. And I was invited here last March, and things worked out. So I negotiated for a start date here in January so that I could finish up some of the work that I was doing previously in California, and just basically got here back in January. So really happy to start.
Well, congratulations on the new job. Yeah. Glad you're here.
Thank you.
My question for you is, what is an integrative physiologist who focuses in environmental science?
That's a very good question. A lot of times in some of the seminars with my students, I love to give that definition up front. So integrative for me means a couple of different things. The first big one is I really love to study animals and animal function. I'm a physiologist by training, and I do so over a number of different hierarchical levels. So meaning I look at how does the animal function at the whole organismal level? How does the animal function, say, at the tissue level or organs, the heart, the respiratory system? I look at how are cells responding to environmental stressors, and also even to the molecular level. So by integrative, it means whole organismal all the way down to cellular and molecular level in the way that I study what I do. And then also by integrative, I also mean you have to take a bigger context to the physiology. And by that, I mean you got to consider things such as animal behavior. How does that play a part in a role in intersecting with how an organism functions in its environment? It can also mean an ecological context. What's happening within the environment? What's happening with other interactions, whether it's biotic, different fish species interacting with one another, predator-prey relationships, food availability, etc. So you're really extending. You might be thinking about fluid flow. If it's out here within, say, the intertidal, how is wave action influencing the animals that you find within the intertidal? Things like that. So it's through a lot of subdisciplines, I guess is what I'm saying. And I really love connecting that with my work, and I really partake in a lot of collaborative efforts to do this because I can't do all that. So I need really good partners to help me in understanding that with the physiology.
So really, they were looking for somebody who's not afraid to tackle these questions from every angle, from the organism to the cell, looking at the environment, the biotic factors, the abiotic factors, but studying physiology in that lens.
Yeah, I think so. My sense was that's what they wanted, and it clearly worked out. So I think it's a great fit for College of Charleston and for myself.
Good.
That's awesome. So how did you get into this field? Like, when you were a little kid, were you like, oh man, I really want to research and whatever when I grow up? Or how did you get to this point?
Well, I know a lot of students, high school, undergrads, it takes them a long time to figure out exactly what they want to do, what path they want to be on. It wasn't that way for me. So I think I first uttered the words, I want to be a marine biologist by the time I was in 7th grade. And I can remember it distinctly. I had one amazing science teacher in seventh grade, and her name was Sarah Jones, and she had actually taught my parents, and
Wow.
she retired the year after I had her. But my learning experience throughout her class in that seventh grade science class was just amazing. It was a cross between Jane Goodall and Jacques Cousteau.
Wow.
So I sort of thought I'd be a hybrid of the two. And then throw in David Attenborough for a twist with just the narrative.
Yeah.
So I knew for a long time I was really attracted to water, being around water. I was into boating when I was very young, a lot of fishing. So that's a big theme in studying fish is the main animals that I love to look at. So it just really hit me like super early. And then that sort of just kind of progressed as I went along. From high school, I went to my undergrad institution and got a degree, dual degree, marine science and biology. So that marine passion was really being nurtured throughout my undergrad. And then I'd say in terms of what I really wanted to do is how do animals work? How do they function beyond just what we see on the outside? Really took off late in my undergraduate years. And that was what I ultimately ended up pursuing in grad school. So I went to the University of Maine, where I started studying the physiology of Antarctic fish and looking at ways that they've adapted to live in such a frigidly cold environment, to future kind of considerations of looking at today. What is going to be the impact of a warming environment in such a frigidly cold place? We hear so much, whether it's the North Pole and the polar bears and loss of sea ice, to all the different ramifications for the warming that we're seeing on the planet. But these animals particularly are being impacted within the Antarctic and the Southern Ocean. So I became really enamored with that. So I guess that's a long-winded answer, but my path has gone for quite some time and just being really, really passionate about what I'm doing today. So it's a dream come true.
Yeah. So with your research on these Antarctic fish, did you actually travel to Antarctica?
I did. And funny enough, I never thought when I was interviewing for the graduate student position that I would be going to Antarctica, even though I knew my graduate advisor studied Antarctic fish. I thought somehow they would come to us in Maine, I guess.
Okay.
So no, it was fantastic. I spent probably the better part of 6 months of my life in Antarctica over 3 different field seasons. Starting in 2005 was my first exposure to the ice, as we call it.
Yeah.
Caught the polar passion and went back again 2007 and 2009. And then I've also been collaborating with some colleagues since that time with samples that we brought back and frozen tissues and et cetera. So it was...
What was that like?
That had to be amazing.
It was. And it's funny because some people, when you first tell them, oh, I was in Antarctica, you get one of 2 responses. It's either what you just stated, oh, that had to be amazing, or why would you want to go there?
Is there anything there?
It's so cold. And yeah, it's a frozen wasteland. It was spectacular. It was kind of love at first sight. From the time you cross the Drake Passage, which is one of the biggest waterways on our planet, circling around Antarctica, and you make it to the peninsula. So that's where I did my work, was Palmer Station. It's the smallest of the 3 U.S. research bases. We have McMurdo, South Pole, and Palmer. Palmer, I think, is the best in my opinion. It's very small. Only 40 people can actually reside there at any one time. A couple small buildings, but you're right on the water. You have the glaciers. You have all the wildlife, the whales, the seals, the penguins, the seabirds. It's just utterly amazing. When you first see your first iceberg crossing the Drake Passage, it's stunning. And then by the time...
I can only imagine. Sign me up.
Yeah, it's an amazing experience. I can't say enough about it.
Yeah.
People, surprisingly, you can actually get there other than being a scientist nowadays. So there are cruise ships that go to the peninsula. So some people pay actually big bucks to do this. But I was fortunate. I actually got to go as a scientist and step foot on the continent.
That's amazing. So while you were in this amazing field station out in Antarctica, what was the group of organisms that you were looking at? What was kind of the research that you were doing out there?
Okay, great. So my work was looking at a huge group, a suborder of fishes called the notothenioids. They are a group that's over 100 different species, highly endemic to the area, so not many coming from different latitudes, but native basically to the Southern Ocean. And we were really interested taking a comparative perspective. So in addition to calling myself an integrative physiologist, I really take a comparative physiology approach. So looking at species that are really closely related to one another and then kind of teasing apart the nuances of what makes them different and how they work, basically how they've devised different strategies to solve the same problem. And that is a lot of fun for me. And that's a big theme within my work. So part of it was in looking at some of the mechanisms, particularly their cardiovascular systems. I do a lot of cardiovascular / respiration type physiology work. But then toward the latter part of my time in Antarctica, started becoming really interested in the effects of temperature on these animals. So they're living in such a frigidly cold environment at this constant chronic cold temperature that doesn't vary very much. I mean, you're talking within a degree or two really most times, or a couple degrees seasonally. And the water temperature is typically in the winter, it can be down to the freezing point of seawater, minus, like 1.9 degrees Celsius. And so living in such, we would call that a stenothermal environment. So meaning a very narrow temperature range that all the organisms that live there have adapted basically over millions and millions of years to be able to live in that kind of environmental condition. So became really interested in what does this mean with warming that we see on the planet today. Particularly the Western Antarctic Peninsula region is one of the most rapidly warming places on our planet. And what does that mean then? Because if you think about it, it may not seem like a lot if you think about, well, what does it mean for a 1/10 of a degree to go up? And that's some of the rates that we see. But if you think about in 10, 20 years, if we go up even by a degree, 1 degree can mean a change from a frozen state to a melting state when you're around that freezing point, right? So,
Right.
And in physiological terms, a 1 degree change in temperature can have really big implications for organismal function. So we started looking at the effects of temperature and what did that mean for this group of fish and how susceptible might they be to these small changes or even big changes in temperature that we're seeing.
So basically, you were looking at this environment that has spent a very, very long time in a very narrow range of temperatures. And we're seeing and predicting that that temperature range is going to change and has already started to change and will continue to change in the future. And you were kind of looking at how are these organisms going to respond as an environmental condition, warming, happens, because we know it's coming down the pipeline, right?
Correct. Yeah, that's a really good summary. And we see changes now already at the ecosystem level, whether it's from a recession with the glaciers going back, even my time at Palmer Station, and over the years, you see how the glacier behind the station has receded over years. You have much more massive icebergs coming off and calving from the main part of the continent. And we see it through the food web, changes, whether it's with the krill, the algae, the cycling, all the way to the penguins, to pretty much right throughout the food web. So we're seeing those sorts of community changes happening already.
Wow.
So with these ice fish that you were studying, what exactly did you learn from them or about them?
Yeah. So, well, the ice fish, so I'll step back just a second. So within the entire suborder of the notothenioids, there's a really special family of fishes within this order. They're called the Conichthyides, or Conichthyidae is the family name, and they're commonly called ice fishes. So when we refer to ice fishes, it's actually meaning the family, which is comprised of 16 species within the family. And what's incredibly unique about this group is that every species within the family lacks an oxygen binding protein called hemoglobin. So without hemoglobin in their blood, they look white-blooded. So it's the only adult vertebrate animal that we have on our planet that this is the case, which is amazing. It's been tried to be reproduced in the lab genetically with knockout mice. You can't do this. No other organism survives without hemoglobin in the blood. But this particular group, these 16 species of ice fishes, have figured out a way how to do that. And it's stunning. The first time I saw one and opened it up, I looked at the gills, and I've been a fisherwoman from early on with my dad. And I remember the first time he showed me a fish, and fish bleed red. It's like you see red. And it was just amazing when you see your first ice fish and you see these translucent, opaque looking gills, and the same for the blood. It's basically this opaque, clear looking blood. Wow. So they figured out how to do this without hemoglobin in the blood. And they have these really amazing cardiovascular modifications. So my big interest in the cardiovascular system, they have really big hearts. They have really wide diameter blood vessels, a lot of them. The vascular density is really high, and they have a lot of blood. So they do have oxygen in the blood. It's carried just in solution, but it's very, very little compared to if they had the hemoglobin. So they've remodeled their plumbing system. You can think about that. It's so cool. If you look at an ice fish compared to one of the red-blooded species there, same size, their hearts are just so much bigger. It's just they've completely figured out a new plumbing system to achieve the goal, which is still to get oxygen to your cells, right? You have to get oxygen to your tissues and to your cells to survive. And they've figured out a completely different strategy of how to do this without the hemoglobin in the blood.
It's just insane to me that there's a group of vertebrate organisms that have completely redefined the vascular system in their group as we know it, because kind of one of the major points of the human or vertebrate vascular system, the heart and the veins and the arteries, is transporting oxygen to our cells so that we can undergo aerobic respiration, right?
Correct. Yeah.
And they said, I don't need hemoglobin, and completely reworked it. And that's amazing. Has there been any look to see what kind of benefit that may confer? Obviously, there are organisms in this environment that do use hemoglobin, and this group of fish do not use hemoglobin and had to make some pretty radical physiological changes in response to that. What might that do to help ice fish as opposed to other organisms?
Oh, good question. This is actually one that's still open for debate within the field for certain. But one we often come back to, if you think about blood and you think about hemoglobin in the blood, and we have, a number of red blood cells that's comprised within our whole blood, which we can look at that and call it hematocrit, the number of red blood cells in whole blood. If you reduce that red blood cell count, okay, and you have a lower hematocrit, your blood is basically going to be, let's say, less sticky, less viscous, okay? So one potential benefit is by reducing that blood viscosity, that stickiness, it's going to have some sort of energetic advantage. It's easier to pump. You can think of it that way. If something is pumping something gooey and viscous versus something that's not in that condition. So it becomes a lot easier. And at colder temperatures, you actually see, even with temperate fishes, seasonally, you will see, say, winter is coming on, you might see a reduction in hematocrit throughout the winter months because, with colder temperature, it's going to slow things down, etc. So there's lots of ways that we sort of see that, but never have we seen an evolution with the complete loss of the hemoglobin. So for the Antarctic ice fishes, this was a complete deletion. So it was an event basically that happened in time that was a complete deletion of the hemoglobin. There's only a slight remnant left that is non-functional within the genome now.
That's absolutely insane. That's one of the coolest evolutionary stories I've ever heard in my life.
Yeah.
Yeah. But in terms of just back to the whole, is there an advantage, et cetera, one of the things, and this is why we're so interested in looking at the warming effects, because as warming happens, there's going to be, number one, there's less oxygen in the Southern Ocean waters because it's inversely correlated, temperature is inversely correlated with oxygen content. And the animal's metabolism, as we warm up, just like for us, metabolic rates are going to go up. So you have more of a demand. You need more oxygen, but yet it's going to be this conundrum in the environment where the oxygen level is going down. So that whole idea of, meeting oxygen supply and demand is eventually going to reach sort of impasse where they may not be able to do that for those reasons. And the other thing, so this plumbing, this remodeled plumbing system that I'm talking about, it's not exactly energetically cheap to run this system. So you can think about this like anything that you would do mechanically. If there's more blood vessels, there's a bigger blood volume, there's a bigger heart, the very pump. It's going to take more energy to basically run that system than a smaller system. So there's a lot of debate on this. They basically get by with it where they live because of the unique conditions that they have. In another environment where their metabolism goes up and they need more, it's going to be a problem. So it's basically their sort of adaptive response, if you will, or the evolution biologists would call me out on that one.
Yeah.
We might actually say disaptive type of response. At the worst, it's basically a neutral mutation is what we're looking at. So meaning the animal can still survive and exist in its current conditions with this, but we might not necessarily call it advantageous. That's a very loaded word for us to use with the ice fish.
Fair enough.
Yeah. I'll be really interested to see kind of as that research continues, what else you find out and where that takes you. That'll be really interesting.
Yeah, I'm excited. I don't have anything on the agenda immediately in terms of my funding, but I'm still working with different collaborators and setting my sights a little bit to the future here and what are the next really important questions to ask and to continue the work, particularly with the thermal tolerance and then also the work with the ice fishes and their really amazing cardiovascular systems.
Well, I can imagine that there aren't so many ice fish in Charleston waters around here. So what is some of the current research that you're doing here at College of Charleston?
Yes. So, not quite like the ice fishes, but there's a equally unique group of fishes here as well. I seem to really love to delve into species that are, closely related, but a cool family type of group. So there's the cyanids here, which is a huge group, and it's one that comprises some of the big commercial and recreational species such as red drum, black drum, and then also one that I'm really interested in studying, which is the spotted sea trout, which is kind of an anomaly. It is not really a trout per se. I grew up fishing for trout. They belong to the Salmonidae family, so they are not a salmonid. They are a cyanid. But the spotted sea trout are kind of unique around here, and they seem to have a cold sensitivity. Meaning, lots of times we've seen winter fish kills where temperature drops here in Charleston Harbor or the surrounding area, and you have potentially large kills that occur with a dropping temperature. So this is kind of a flip side for me. The Antarctic fish I was looking at, warming sensitivity, and now I'm kind of switching gears a little bit and really kind of fascinated with looking at what is it that's defining these cold limits for these animals. Because with global change, there's areas, we typically talk about warming, but the fact is there's areas too where we see these changing environmental patterns where sometimes cold can also play a big role. I mean, the biggest thing is we see just shifting temperature regimes everywhere. And so I'm really interested in looking at the cold tolerance of this particular species. So.
Absolutely. And I think that's a really important point to kind of hit is that global warming, yes, the Earth is absorbing more energy as a whole. And so we're seeing the global climate rise slowly, but just because the average temperature is rising, that doesn't mean that the extreme temperatures are always going to be warmer. It's really, you put more energy into the system, and that's going to destabilize the system. So we'll see parts of the Earth that are getting hotter than they ever were before, but at the same time, we'll also see extreme cold spots where there's shifts across the seasons that are much colder than they used to be. It's more of a destabilizing than an overall trend towards warmer.
Absolutely. That's a fantastic way to put it. And I think that's one of the things that people lose sight of when we talk about global change and global warming per se. But it really is that destabilization and big differences across the globe. And I think it's going to be really important in looking at both ends of the spectrum and looking at this variability, which for animals that live in constant chronic habitats, such as like the Antarctic fish, they're not used to having some of this variability. And there's many animals within, nature that are going to fall into that sort of category. So I think defining, and looking at the different counteracting effects and where that's going to occur, who's going to be most impacted. One of the things in my field we like to talk about is who are going to be the winners and losers.
Right.
Unfortunately, the fact is there are going to be some species that do much better than others, and there's going to be some that aren't going to fare very well in light of some of the changes that our planet is seeing. So I think it's important, in terms, if we kind of just jump forward and think about conservation, especially fisheries management, how do we best help our resources of what we have in the ocean? We really need to know and be making some of these predictions and what's going to happen at the species level. What does that mean in the community, the population for these species, so that we can better prepare for the future and also protect our resources in the ocean.
Yeah. Now, how exactly are you studying these fish? Obviously, you have a lab here. Are you doing a lot of that research in your lab, or are you out in the field a lot?
Oh, great. So, still moving in, so I'm not quite in the lab yet, but I'm really looking forward to getting my full lab up and going. But so it's going to be a combination of both work within my lab, so sort of at the classic lab bench. We do have holding aquaria. There will be animals that I will be bringing in, doing different environmental exposure work within the lab, and then mechanistically looking at how they're responding to some of these changes, such as temperature, dissolved oxygen, potentially salinity, you name it, whatever factor I really want to look at. But then importantly, in the last few years, I've had a major interest and push to connect my work that I do in the lab to that in the field. And the reason for this is I feel it's really difficult. We can look at the mechanistic level and we can define individual variables or multi-variables with some of these cross-variable experiments that we do within the lab. But until you go out into the field, it's really hard to put together the full picture of how an organism is going to be responding when you have so many different factors that are really imparting, on what they're doing, where they're going, how they're behaving, how they're functioning, etc. So I've really been trying to see newer techniques. What can I do to sort of advance the field forward in bridging that gap between the lab and the field, so that we get a clearer understanding of what's really happening with environmental change when you put all these factors into play: temperature, oxygen, pH, food availability, currents, predator-prey relationships, etc. So a lot of my work has been in making that push into the field.
That's great.
Yeah, this has been awesome. I really love how you're examining how individuals and species react to temperature change in different ways, and trying to suss out how one species in a closely related group or another species, how they react differently and what that means as far as management strategies. If we know that an organism has these biomarkers, these particular suits of adaptations, can we generalize that? Can we say, well, this is another species that we've looked at that has some of these same biomarkers, but we haven't done as much research on it, and maybe we can sort of expand our management strategies in a way like that. This is some really cool work that you're doing here.
Yeah, thank you. I think so.
I suppose that's probably why you devoted your life to it.
Yes, absolutely.
Well, thank you so much for being here. This has been a great conversation.
Thank you.
Thanks for being on the show.
Thank you. It was my pleasure. Thanks for having me.
Thanks for listening to this episode of Conservation Connection.
If you enjoyed our podcast, go ahead and subscribe to make sure you catch every episode that we post.
We would love to hear from you. If you want to reach out to us, go to our website, lastchanceendeavors.com/contact, and shoot us an email.
If you're a teacher and you'd like to use this episode in your classroom, get in touch with us.
We'll provide you with state and national standard tie-ins and additional materials to help you guide a class discussion.
Thanks again for listening, and don't forget to tune in next week.